Good afternoon, ladies and gentlemen, and welcome to the E-Therapeutics Interim Results Investor Presentation. Throughout this presentation, investors will be in listen-only mode. Questions are encouraged and can be submitted at any time using the Q&A tab situated on the right-hand corner of your screen. Please just simply type in your question and press send. The company may not be in a position to answer every question it receives during the meeting itself. However, the company will review all questions submitted today and publish responses where it's appropriate to do so. These will be available via your Investor Meet Company dashboard and will notify you by email when they're ready for your review. I'd also like to remind you that this presentation is being recorded. Before we begin, we would like to submit the following poll, and if you would give that your kind attention, we would be most grateful. And I'd now like to hand over, if I may, to Ali Mortazavi, CEO from E-Therapeutics. Good afternoon, Ali.
Good afternoon, Mark, and thank you for having me on. Okay, thank you everybody for joining me this afternoon. I've done a more bespoke presentation, as there's I think quite a few people that are maybe new to the story. You can get a copy of the presentation that says that the link is at the bottom of our RNS today or on our website. Nothing price sensitive has been left out in this presentation, but I think this is a better introduction to the company. Okay, so E-Therapeutics is a computational biology company, and really we are a play on the kind of convergence of technology and the life sciences space that really is an inevitability, and the two fields are kind of getting closer and migrating closer and closer together. I will have just a very quick, glance through the very unremarkable kind of results, just to say that, the 6 month ended July 21, we had 31.6 million of net cash, a healthy cash balance after a capital raise of 22.5 million. Obviously, that's been, really instrumental in, kind of for us to be able to implement our plans, and the kind of subscribing shareholders were the very, very highest quality, and very, very happy to have them on board. Okay, so what does E-Therapeutics do? What was my interest in E-Therapeutics, if you like? Why did I join the company? In very sort of high level terms, what E-Therapeutics has is a in silico, or a laboratory within a computer. Kind of what does that mean? That basically means that we can model and analyze biology within a computer. And a kind of the starting point is, well, how do we do that? How does that evolve? What is a model? The starting point for almost anything relating to computation is data. So our input data and the data sets that we've generated are critical for us to be able to sort of construct models of biology. So we have what are called protein-to-protein interaction databases, biological pathway databases. We have the most complete library of something in the region of 15 million small molecule compounds. All the approved drugs are in there. These compounds have all the kind of chemical signatures next to them. We also have, a particularly important function. All the data sets that we use range between, in terms of quality, to very high quality, very high annotations, middling, and sometimes poor. And in the kind of middling and poor region, where we really have kind of invested a lot of time and capital in, is in machine learning algorithms, which really fill in the gaps in trying to predict where there's missing data, what should the data be in there. And that's a kind of critical function of really being able to get data, make it as complete as possible, and curate it for our own needs. I should say that, this is really what I would say kind of man plus machine venture. We're not at the stage where anybody really is able, or any algorithm is able to sort of say, press a button, computer gives answer. So, we have a very skilled set of staff who are, very kind of multifaceted computational biologists, who are also cell biologists, who are medics, and they're very integrally integrated into the whole system, into the whole kind of data science, the algorithmic part. But importantly, what the skill sets that we have are not just at a computational end. We have a lot of experience in the drug discovery and drug development world, so that we can sort of really try and integrate these two fields together. So, what do I mean by creating models of biology? I think the next slide will be a little bit scary on first sight, but if we go a little bit slowly, we can really work out that it's maybe a little bit more intuitive than it looks. So the circle is a very, very simple schematic of one cell. This you can think of as the nucleus in one cell. And here we just listed just a very few select what we call biological functions within these cells. It really doesn't matter what they are. There are actually thousands, tens of thousands of biological functions within these cells. But kind of one way to sort of separate them and segregate them is if we just follow the arrow for trophic signaling, for example, there's a network of genes really, which is, if you like, embedded within that cell. And that's the network of genes, which is really related to trophic signaling. For example, endocytosis will have a different one. Think about it as the blue network. Or if you like, endocytosis will be the green network. So within cells, biological functions are actually networks of genes and proteins. And they're kind of, if you can sort of almost visualize them kind of sitting there. And, when you start getting into the tens of thousands of things, the number of different sub-networks within these cells is obviously getting to incredibly large numbers. So one of our kind of primary functions is the ability to construct these networks in a computer using our protein to protein or gene databases. We can take a biological process like, say, trophic signaling. If we want something which is a bit more sort of commonplace, a biological kind of process could be hyperinflammation because of COVID-19. That'll be a process within a cell. We can basically, within a computer, stitch these networks up because we know the kind of functions of each individual node. That forms a model. We kind of almost take that network out of the cell. And then there's a kind of process where if this model is a true representation of what's going on in real cells, we can start analyzing what to do with this biological process. So taking hyperinflammation in COVID, obviously that's something that has a potentially catastrophic outcome in the ITU, etc. There's a huge amount of work going on is how do we stop certain people from having this kind of hyperinflammation process. So we would model a process like that. Once we've modeled it, we now begin to think of how we can sort of disrupt it. So imagine if the kind of hyperinflammation was the blue network. One of the kind of things that we can do is think, okay, which node in the network can we try and attack or remove via our small molecule compound database? You think about it almost like playing a game of Space Invaders, if you like. Compound number one, remember we've got 15 million compounds, attacks this node over here. And literally you iteratively go through it, obviously because it's in a computer, it's extremely fast. Maybe Mark, if you could show video number one, which I'm pretty sure the whole system is going to go wrong. So this is an example of attacking the wrong node with a small molecule compound. So what you can see here is, a few of the kind of circles are flying away. But if this was a model of hyperinflammation, this small molecule compound, which has just been tested against this, really hasn't had much effect on this network. So if we want to say dissolve this network to stop hyperinflammation, the one particular node and that small molecule is the wrong place to start doing that. If you show video number two, Mark, you'll see a much more potent small molecule attacking a different place in the network. And now you can see that's a much more profound effect. And, one of the reasons, for example, for potential drug failures that you see in such a high failure rate is if you don't look at kind of biology in this kind of networks and network form, attacking networks in the wrong places can have completely different consequences. And that's because these biological networks are very resilient to attack. They're kind of designed by evolution not to be disintegrated very fast. They do, however, have weak spots, and they do have, if you like, what we call hidden players. But in order to find those, you really have to do the computational work to do that. So it's a kind of very high level view of, we take our computational biology platform, lots of data feeds, lots of machine learning to enhance all the data feeds. We model biology using network biology, kind of what's called systems biology. We are able, because it's in a computer, to test millions of interventions, different nodes, attack different places. There's lots of other different things that we can do, which is probably beyond the scope of this talk. But that is the power of being able to do fast throughput processing and analysis in a computer. And that is what is impossible to do in, cell-based assays in animals or humans, because obviously, that kind of process is impossible to be testing millions of different, sometimes tens and hundreds of millions of different sort of strategies. Okay, so I'll give a sort of brief overview of our computational platform and the kind of data sets that come with it. And today we put out a very important announcement in RNAI, which kind of may seem very almost disparate to what I've been talking about, but I'm taking a little step back to when I first joined E-Therapeutics. What interested me in this company was I believe that the computational approach, our networks approach, I believe that computational power, the unlimited resource space, what's called dynamic compute that we can have via Amazon, we can hire unlimited computational power for days at a time here and there. I believe that there is the capability there to really change the entire process of drug discovery. So on the left-hand side, what you'll see is some competitors of ours. Some of them you may have heard, US companies like Recursion or Benevolent. I'm not going to go and differentiate between their platforms and ours, computational chemistry and biology. On the right-hand side, what you see is what's called the RNAI world, and I happen to have experience in both of these camps. So I was a CEO of Silence Therapeutics on the RNAI world, and obviously now joining kind of E-Therapeutics, got a big kind of, if you like, exposure to the whole world of computational biology. And it was pretty noticeable to me as soon as I joined E-Therapeutics that there's a kind of asymmetry going on. Obviously, on the left-hand side here in the computational world, you've got almost kind of warp speed, unlimited speed of processing. But at the end of the day, as I said earlier, there is still no way that a computer can ultimately ever say to you, this is a good drug, this is a good drug target, and give you answers like that. We still, even in this convergence theme, we're still at the point where there has to be a hybrid approach. You can gain big advantages via computational approaches, but whatever your outputs, you have to actually go and test them in a laboratory, in an animal, and ultimately, if you want to make a drug, in a person. In order to do that, you've got to make a drug. And in this kind of circle, primarily the drug of choice is what are called small molecules. This is where E-Therapeutics was in this box. And what was striking to me was, let's say, even the best outputs from the computational platforms, remember, these are running at very, very high speed, lots and lots of different ideas. If you wanted to make a drug in small molecules, it would take you 4 years and about $4 million. Actually, that's an understatement. It could be a lot longer than that. So you've got this kind of asymmetry of speed. You've got something which is processing God knows how much information, really relevant information. But if it's going to take you 4 years and $4 million just to make a drug, let alone test it and all the other things that have to happen, you're suddenly going extremely fast to unbelievably slow. The flip side of that is my experience in the RNAI world and at Silence was that we could make our drug in 6 months and about $500,000, and actually probably less than that, depending on your definition, but 3 or 4 months, even $300,000 or $400,000. So, my kind of vision when I joined was, could I get E-Therapeutics in this intersection where we could kind of capitalize on this and capitalize on at least as fast as we could go, so that if we generated ideas, that we could prosecute them as quickly as possible. Obviously, this is a materially different timeline and cost to each other. So kind of by a little bit of background information, why are RNA molecules, why are these things so fast compared to, let's say, small molecules? And that's because the drug is really what's called an information molecule. And kind of maybe the best example that is pertinent to everybody is, think about the vaccines that most of us have taken, Moderna or Pfizer-BioNTech. How did that work is once the spike protein was kind of the genetic sequence of it was known, the vaccines could be designed in a computer in literally days. So what happened is you took the sequence, then you went and made the vaccine extremely quickly. Upon injection, your body read that sequence and the immune system kind of kicked in. And these are information molecules because the drugs are actually a genetic sequence. In a kind of similar vein, the RNAI platform is very kind of similar in terms of that information molecule. So there are around 10,000 genes, let's say, in the liver. All these genes have a particular sequence. If you want to make a drug against any of those genes, you're really trying to find a sequence which is almost the opposite to the sequence you want to target. So think about 10,000 genes. You can have a short interfering RNA number 1. Gene number 2, that's RNA number 2. So kind of if you visualize a shelf, a rack full of sort of 10,000 different interfering RNAs, genetic sequences, against every single gene in the liver. So again, I'm still in between worlds. I've seen this work in a previous life, had a lot of experience with it. I also knew that, in the world of RNAI, you could deliver these kind of information molecules to liver cells, and very specifically to liver cells. So that was kind of a big advantage. But to go from here and to get to, if you like, this intersection and actually to execute that took an unbelievable kind of amount of really work during almost lockdown. We started a year ago, looked into the hundreds, maybe thousands of patents. This is a very, very protected area. Things that have taken people, other companies, billions of dollars in years to be able to come up with technologies that worked. So it wasn't an easy ask, and despite all the experience, you to find a way through the IP, the actual chemistry, etc., was pretty daunting. But the goal of having to be here was particularly important to this company and to us, to me, because this was just not an option to be in small molecules. This go very fast, go very slow. It's just not a strategy that we could do. Okay. So this really, a couple of slides, is really about our second announcement today about what we call the benchmarking or characterization of our RNA platform. So as I said, a year ago was really the sort of desktop work, patent, reading so many patents, lots of patent attorneys to say, okay, can we kind of navigate our way around that? And in around February, we decided on what we say 8 designs and what we call kind of different delivery systems. So when I say designs, think about this as the drug, siRNA, it's called the short interfering RNA. So we designed 8 different versions of this. Versions of them will be, for example, we modified, without getting into too much detail, some of the positions and bases. Sometimes we moved the delivery system to other locations. So at least in kind of conceptually, we said, right, we think these 8 different designs has the best chance of being potentially successful and giving us a technology platform. And we set ourselves a pretty high hurdle. We said, look, we're going to choose 3 targets in the liver, 3 genes in the liver. 2 of these genes we knew that we could compare against the kind of state-of-the-art data. So they're approved drugs. We knew all the data that we needed to beat. There's a 3rd target, which is a very kind of high-profile target. So we had set up experiments where the kind of object of the exercise was at least one of these 8 designs had to be at least as good as the very best data that was out there. Very, very high hurdle. Again, think about the kind of spent capital, et cetera, and the time we're talking about. February, I think we finished the experiments literally a couple of weeks ago, maybe a week ago. So in the space of 9 months, we kind of really managed to score as well as we possibly could. We now have in 3 different gene targets comparable data across species in mice and rodents and non-human primates, and our best designs are consistently the same across the kind of 3 targets. We filed 11 new patent applications to protect our positions. And as we said today, we're showing at least equivalence, I think, to the very best platforms that are out there and really managed to enter a multi-billion-dollar kind of industry during kind of lockdown. And I'm very proud of the people who kind of worked on this. So, and sometimes I think without lockdown and that ability to really focus on something, maybe we wouldn't have been able to do it as quickly as this. But it is the best case scenario that we could come up with. And without question, it's a material success for the company. So if I'm jumping a few slides, I apologize, but suddenly this dream or vision, whichever you want to call it, is now very much a reality, that we have all the best of computation and all the possible maximal advantages of a drug platform to go with it. Okay, so I won't go through all the data, and as I said, you can find it on our website or on the RNS at the bottom of the page. But just very slowly and quickly, I wanted to just sort of give you an example of what I meant by designs. So by designs, if you think of this as design number 1, ETX019, design number 2, design number 3, 4, 5, 6, 7, 8. So 8 different designs, as I said. And this is target Y, which we haven't said what it is. So think a good score here is for this from 1 to go as low as possible. So as you can see, the red and the orange and pink are doing particularly well here. Some of the other kind of designs, very variable. This one particularly poor. To give you an idea of, there's a very big difference between designing things in a computer or intellectually and knowing the results of an experiment. I kind of thought probably if I had to have a guess that this one would do very well, but one of the poorest ones. In any case, ETX 23 and 24, which are 2 specific designs, are doing very well. And I guess on the right-hand side is really the kind of equivalence at being as good as the world-leading platforms kind of data that's really very important. So again, going very low is important. What this is saying is there's an injection happening here. This particular gene, you can see that it's losing its function over 14 days. The red line is our line. At 28 days, it's lost 90% of its function, and that's what we want because this gene is probably causing a disease. And then you can see a slow recovery, but still even here at 50% function, it's therapeutically relevant. And, we have nearly 3 months of activity with a single injection. And this was compared against, as I said, the state-of-the-art comparator. So remember, lower is better, so this gap is good. And as you can see, we're at least as good, if not slightly better here, and matching and equivalent here. It's amazing how kind of, so many few million pounds and so many data slides can actually come down to almost one very simple graphic. But this very one simple graphic, I think, allows us to really say that, we have a technology now that can do what we call silencing, or stopping a gene function of almost any gene in the liver with 1 injection and probably up to 3 months of duration of activity. And that's as good as the state-of-the-art systems that are out there. Okay, so how does that sort of go with the computational platform? Well, the first thing is we are able to really make our computation much more specific. So as I said earlier, the drug technology is very unique in that the delivery system takes the RNA only into one cell type. If you want the liver, and hepatocytes are a big part of the liver. So what happened is now we can run the parallel strategy. We extracted all the data which is hepatocyte cell type related from our general platform. Everything has gone into a kind of new sort of bespoke and focused group. I think kind of what that does, if you think about, human biology in the system, is we have now managed to, let's call it, narrow down to one cell type. We know that our drug always gets there, but we're also narrowing down all the biology to that cell type. So I've used the word narrow because, for narrow, you can replace specific, one of the two. Actually, it's far from narrow because the hepatocytes represent such a big percentage of the cells in the liver. As I said, there are 10,000 genes or proteins out there. And the liver is one of the most active organs in the human body. We're not just talking about liver diseases here. Genes that are basically made in hepatocytes are responsible for liver diseases, a lot of cardiovascular diseases, blood disorders such as hemophilia. So actually this narrowing that I talked about is still a very gigantic biological space, an unexplored biological space for our company. Okay, so in parallel and slightly on spec, because we didn't know the results of the drug platform to come, we developed a very hepatocyte-specific data strategy and what's called a knowledge graph. And what is a knowledge graph is ultimately, again, it starts to look like one of those funny networks, a kind of hairball or mesh of information, really. So we have extracted using kind of software called natural language processing, which just goes out and just imports as much relevant information and data to hepatocytes. We kind of importing almost every biological experiment that's ever been done. We've got all the chemistry related to that, all kind of pattern searches. We can look at any kind of what's called omics data. Any data which has got any relevance to hepatocyte biology is being imported into our knowledge graph. And, this is an evolving system. It's not something that's ever going to stop, really. It kind of gets bigger and bigger and bigger. But, the plan, and, I think we're well on the way there, is for this resource to become, the place for biology and hepatocytes, where we can, use it for partnering or for our own kind of purposes. What's kind of interesting is the kind of size of the information here is obviously getting larger and larger. Our Amazon spend and compute is going up exponentially. There is a way, for example, look, this is kind of zooming into one sort of bit of the knowledge graph. So if you want to do a search, if you like, by a gene and a drug and a disease, you can get to kind of very local sort of places within this, look at it yourself if you want, manually. But really, there's so much information that, ultimately our goal is to have, if you like, kind of machine learning algorithms which are constantly scanning and surveying and have strategies to find and extract best correlated data to each other, to anything that can give us a kind of advantage in understanding certain aspects of the biology that we're interested in doing. Okay, so in terms of kind of value inflection points and business models, there are many ways to sort of slice and dice this. If we start with a core platform, RNAI platform, which is the data slide that you saw earlier, the ability to be able to, as I said, remember, silence those genes or knock out a gene function of the 10,000 genes. We can do deals here. A template deal is one of our rivals called Dicerna with Novo Nordisk. Novo Nordisk in 2019, they reserved 30 genes out of those 10,000 in the liver and paid Dicerna $175 million up front plus milestone payments. So these are exclusively owned by Novo Nordisk, can't be prosecuted by Dicerna. What Novo Nordisk are paying for is the ability to use their platform, the same as the platform that I've shown you. And obviously a very big differentiator from us from any kind of RNAi competitor is this dedicated resource, the computational platform, algorithms, and hepatocyte-specific data. We could do deals on a kind of purely biology basis, for example, kind of investigating a lot of data sets in what are called regulating RNAs and genomic regulations within hepatocytes. Those are sort of slivers and areas where we could go and do deals within that. So kind of many ways, like I said, to slice and dice it. Kind of ultimately, if we have from our outputs, from our kind of resource that we're building, if we have high conviction targets of our own that we want to go and prosecute, well, then we can sort of, from the go signal, you get into this very fast lane. You can make the drug very quickly. Within 18 months, you've got $1 million. You've got to read out in a relevant disease model whether your drug is working. There's precedent at this point. We're on Amgen Arrowhead. Amgen paid $35 million up front, $21.5 million in equity, plus further milestones for one target and biological data that Arrowhead had generated here. To go even further, if you want to go into phase 1, again, Dicerna with Roche, $200 million up front for one phase 1 HPV with a kind of big billion-dollar milestones. And obviously, you've always got the potential to go further with your assets if you really have a very high conviction and belief in them. So many different areas to de-risk, partner necessary. But I think an important part for us is to be able to use this big timeline advantage. If you add these numbers up, with these timelines, 18 months, depending on what disease, 18 months to 24 months and $1 million, you've got really relevant data there. If you go back to the small molecules, they're still making their molecules and they're still 3 years, 2 years behind, if you like. Okay, so summary and next steps. As we said, we now kind of have the ability to inhibit, silence, reduce any kind of gene function in hepatocytes and rapidly generate drug candidates. Today, we also announced that one of our collaborations away from the RNA side on the general platform side with Galapagos has passed the next hurdle. We expect to, we're kind of final gating structure of the go, no-go at that. We hopefully get an answer in the next few months on that. As I said, the most really specific and extensive hepatocyte data resources being built well on the way. Mentioned the automation and cloud computing changes all the time. You'll be hearing from us in an R&D day in 2022 where the kind of knowledge graph and these algorithms, everything will come out of presentations and be on screens and show how they actually work in practice. And we are very excited by our results today. Thank you very much.
Ali, thank you very much indeed for updating investors this afternoon. Can I please ask investors to continue to submit their questions using the Q&A tab situated on the right-hand corner of your screen? But just while Ali takes a few moments to review those questions submitted already, I'd like to remind you that a recording of this presentation, along with a copy of the slides and the published Q&A, will be accessed via your Investor Meet Company dashboard, and we'll let you know by email when they're ready for your review. I'd also like to remind you that your feedback is important to the company, and immediately after this presentation's ended, you'll be redirected for the opportunity to provide your feedback in order that the company can better understand your views and expectations. Ali, I haven't given you a long while to look at the questions that have come in, but thank you firstly to those investors that have submitted questions. Perhaps, Ali, I could just hand back to you to read out the questions and where appropriate to give a response, and I'll pick up from you. Thank you.
So I'm looking at Michael D, who says, how much of your scientific headcount is outsourced and how do you see this moving forward? Definitions kind of count here. We outsource, like for example, in the RNA characterization, we used kind of very big CROs for that rather than internally. But in terms of the whole computational aspect, everything is internal and full-time staff. Okay. What have we got here? Josh M saying, which would you consider the state-of-the-art platforms that you're comparing ETX24 to? That's a kind of awkward question because obviously we've kept the targets anonymous. We've kept the company names anonymous. But what I would say is we are comparing ourselves to the very biggest and the very best. I think that should answer it. Are there any other partnerships in the short term similar to that with Galapagos that will help your development? Asked Andy. Yes, we've said in our results that we've got multiple interest in doing so. I think what we are in the zone of is I think we could have done smaller deals. We are somewhere between, we don't want to do the smaller deals that take bandwidth. We want to do Galapagos plus deals because that kind of the projects need to be meaningful. Really what they do for us is stress test our systems, our networks in real life pharmaceutical settings. I'm hopeful that we can do similar type deals. Yes. Okay. Some of the other questions are kind of more sort of giving a little bit of forecast stuff, so I can't really do that. And Josh M and David D, I mean, it's just difficult to sort of be able to do those. Peter A asks, how do you safeguard your IP? We use way too many IP attorneys and global patent attorneys, but, kind of very robust system for doing that. And we have a very, very good in-house dedicated IP counsel. So that is, I think, it, Margaret. Good balance sheet there. Yeah, I think that's good.
Absolutely no problem, Ali. And obviously, you can only answer those questions within the confines that of course, as a listed company, you can. So thank you, Ali, for taking the
Thank
time
you.
to respond to those questions. Ali, I'll shortly redirect investors to provide you with feedback. I know investor feedback is important to you. But perhaps before doing so, I could ask you to just have a few closing remarks just to wrap up with.
Oh, you caught me off guard.
Yeah.
I think my closing remarks really kind of ended. I think we really got the best case scenario of the kind of RNA characterization, and in terms of data, the time that it came, the speed at which we've kind of managed to go from literally ground zero to actually being able to show this kind of biological activity in IP is really quite remarkable. It's a really key enabler to really kind of put the jigsaw puzzle together of this kind of hepatocyte computational specificity and the drug specificity. I think now the company's strategy is very, very clear, and I think you will be seeing that, we're going to be kind of very consistent in this very large biological space.
Ali, thank you very much indeed for updating investors this afternoon. Could I please ask investors not to close this session, as you'll now be automatically redirected for the opportunity to provide your feedback in order that the company can better understand your views and expectations. This will only take a few moments to complete, but I'm sure will be greatly valued by the company. On behalf of the management team of E-THERAPEUTICS PLC, I'd like to thank you for attending this afternoon's presentation. That concludes today's session, and good afternoon to you all.